Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Navigating the Complexities of Nash Development, Valuable Insights from a Key Opinion Leader. My name is Andrew Jordan and I'll be your X Talks host for today. Now, today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box. And we'll try to attend to those questions during the Q&A session. This chat box is located in the control panel that's on the right-hand side of your screen. And if you require any assistance, you can contact me at any time by sending a message using that chat panel. And at this time, all participants are in a listen-only mode. And please note that this event will be recorded and made available to you for future download. And a big thanks to Medpace who helped develop the content for this webinar. Medpace is a scientifically driven global full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach. And another thanks goes to Pinnacle Clinical Research. They're dedicated to conducting late-phase clinical trials in the areas of hepatology and gastroenterology, with a special focus on fatty liver disease. They pride themselves on conducting high-quality research as a complement to the medical care that their volunteers receive from their routine care center. Pinnacle has highly skilled board-certified investigators involved with the daily oversight of clinical trials. Their coordinating team consists of clinical research coordinators and research assistants with extensive experience in conducting all phases of clinical trials. Now, I'd like to introduce our speakers for today's webinar. Dr. Piatrashievsky is an internist with a broad background in pharmaceutical research. He has over 19 years of experience in clinical drug development with global expertise in the design and medical oversight of clinical trials in the area of gastroenterology and hepatology, including Nafld and Nash. Piotr is recognized in the field for his contribution to the pioneering work on standardization of central imaging in drug development. And Ms. Jen Harness is a registered dietitian with significant global project management experience as well as clinical experience specializing in metabolic diseases, including diabetes, lipid disorders, obesity, and NASH. She has a strong clinical background with over 13 years in clinical operations at Medpace. And joining Medpace today is a renowned key opinion leader in Nafld and Nash. Dr. Stephen Harrison is from Pinnacle Clinical Research, and Dr. Harrison is board certified in both internal medicine and gastroenterology. He is a peer reviewer for over 20 medical journals and internationally known for studies in hepatitis C and non-alcoholic fatty liver disease, with over 170 peer-reviewed publications in these fields. Dr. Harrison most recently served as colonel in the United States Army. Retiring in 2016, he concluded 20 years of dedicated service to his country. Now, it's with my pleasure to hand the presentation and the mic over to your first speaker for today, and that's Ms. Jen Harness.
Thank you all for joining today. Our agenda covers 3 relevant topics in NASH trials, the first one being recruitment, and then we'll talk a little bit about biomarkers and endpoints. We'll cover these topics through a series of questions that Dr. Kresge and I will pose to Dr. Harrison. So without further ado, let's go ahead and get started with the first one. So Medpace has been working in NASH since 2010 and has experienced the shifts in recruitment strategies. Dr. Harrison, having hands-on experience in recruiting NASH patients over the last decade or so, can you tell us what are the current strategies to successfully recruit NASH trials today?
Yeah, thanks, Jen, and hello to everybody on this Thursday morning. It's good to be here. It's good to be fresh back from the International Liver Congress in Paris, where we learned quite a bit about the field, and probably the biggest takeaway from that is how rapidly this field is moving forward. And you blink your eye and things have changed dramatically. And we'll talk a little bit about what the changes look like, what that means for the field moving forward. But suffice it to say that it's all good. Things are just moving very quickly. So reflecting on the current trends in recruitment, that has been, I think, an area that has been overlooked when we talk about drug development in fatty liver disease. For the longest time, looking back to Pivens and Flint and Golden 505 and all the trials that were early on in this field, it's been a bit of a challenge finding patients for these trials. It's not that they don't exist. There's a plethora of fatty liver patients out there, but part of it has to do with building a network of sites that are focused on fatty liver and that have worked in the field long enough to have a disease education program that expands out into primary care, endocrinology, and gastroenterologists in the community. So I think the first bullet here is finding the right sites that are capable of both recruitment and then also study procedures, and maybe touching on that a bit. So we'll talk in the presentation today about non-invasive procedures that we can do in fatty liver disease to both enrich our patient population for the likelihood of what I would call juicy NASH, or fatty liver disease with significant amounts of inflammation and fibrosis. And that includes both wet or blood-based biomarkers as well as imaging-based biomarkers. And it's been a bit of a challenge finding sites that have both patients, and number two, that are married or wed to radiology sites that are capable of doing some of these latest and greatest imaging procedures, both MRI based and also ultrasound elastography based. And so it's really critical that when you begin to look for sites that you do your due diligence, and quite frankly, this is a little bit more than just a feasibility survey. It's really getting hands-on and beginning to understand what those capabilities are, both for proton density fat fraction with MRI, for multi-parametric MRI, for MR elastography. And again, we'll touch on that a bit more. And maybe another thing with recruitment that's critical is enriching the pool of potential patients. And we do this through two mechanisms that I think are critical to enrolling trials. Number one is disease state awareness, really working with the sites to get out into the community, not just your typical advertising campaign, but really go boots on ground and get into the offices and show physicians what they're missing. Docs may not change their practice based on one double-blind randomized placebo-controlled trial published in Lancet or New England Journal, but they will on one anecdotal experience. And we've seen that in our practice here. If we're able to go into community practices and show them patients that they've treated for years, but they never focused on their liver disease, and we show them that patients right under their nose are actually progressing, then that is very powerful, and that really revs up the recruitment that we get. And ultimately, this comes from what we term a pre-screen strategy. I mean, really focusing on what the trial criteria are, and then kind of mirroring that up with a pre-screen strategy that would allow patients to filter into the trial and that qualify, so that your screen fail rate is low. If you think about screen fail rates for clinical trials, particularly liver biopsy-based trials, 60% of all screen fails are due to liver biopsy. So if we can just move that needle a little bit, dropping the pre-screen rate, the screen fail rate from liver biopsy from 60% even down to 40 or 30%, we dramatically change the whole paradigm of clinical trial enrollment. So I think ultimately it's kind of a combination of a lot of things. It's finding sites that have good ability to do imaging, to do wet biomarkers, and that have the ability to get out into the community and focus on primary care and endocrine as well as other GI docs. And finally, ultimately, that's where we've worked very closely with Medpace in leveraging our site network, which is kind of a combination of a lot of sites that are like Pinnacle, that have central IRBs, that are able to get off the ground quickly, but have a historical track record of being able to find patients that can enroll in trials and minimize screen fail rates at the same time.
Thank you, Stephen. Let's take a deeper dive into the practicalities of Nash trial recruitment. How can differences in epidemiology and patient management paradigms in the US versus Europe be leveraged to improve recruitment? Now, this question has an underlying hypothesis that there are differences in how quickly trials recruit in Europe versus the US. And before I hand over back to Dr. Harrison, let me provide a little context and talk about some data that we pulled from, that is publicly available. We pulled it from Citeline. And this is data on Nash trials which started and completed over the last 3 years. So pretty recent data. The blue bars on the left here, the 5 blue bars depict individual studies which were completed in the US only. And the green bars to the right are individual 4 clinical trials which completed globally, actually, but they had a representation of European sites. And obviously the size of the studies may matter, but if you look at the mean recruitment rates, even after excluding the one outlier, the single study in the US, you can still conclude that there is at least doubling of the rate in recruitment of NASH patients in the US versus European sites. So what is it in the US and not there in Europe that we could potentially change or leverage to bring the European trials up to speed? Stephen, could this be epidemiology?
Yeah, that's a very intriguing question, and one that I think over the next couple of slides we'll try to provide some answers to. I think if you look at this data that's put together by Zoberio Nasi out of Virginia, it's a nice meta-analysis that looked at where we stand with prevalence of fatty liver disease around the world. And these assessments suggest that North America and Europe are pretty much aligned on the prevalence of fatty liver disease as a whole. Now, again, when we think of fatty liver, not all patients with fatty liver can enroll in trials. We're really focusing on those with NASH and some degrees of fibrosis. And I think maybe taking a careful dive into different ethnicities and maybe different demographics might provide some inferences into why there may be differences in demographic enrollment from the US to Europe. And I think, again, this is another meta-analysis more recently published in what we term the Blue Journal, or Clinical Gastroenterology and Hepatology. It's a clinical companion journal to the bigger impact journal Gastroenterology, but a lot of very good data gets published in this journal. The impact factor is quite good. And what you see from this meta-analysis is that doing a little bit deeper dive into some of the demographic data, that Hispanics tend to have more fatty liver than Caucasians, and yet again over African Americans. And if you focus on the Nafld population with NASH, we also see the same trend lines that Hispanics are preferentially afflicted with more advanced stages of disease than Caucasians and African Americans. At least from this data set, if you focus on the NASH population and you look at the degree of fibrosis, there is more alignment here across ethnicities than what maybe I would have previously expected, in that it shows, Whitley, with F2 to F4 disease, there's not a lot of difference, certainly between Hispanics and Caucasians. African Americans trail off a little bit, but maybe even a little bit more than what I would have expected.
And Stephen, in your experience and observation, other than epidemiology and obviously the genetic background, we all know PNLPF3 variants in Hispanics. Is there anything else that could influence the volume of Nafld Nash patients of Hispanic ancestry actually available for clinical research that you can tell us about?
I mean, really, what we know focusing on Hispanics is that they, in addition to the PNPLA3 variant, they just historically they tend to be a population that is enriched with diabetes and also obesity. You know, here in Texas, we have a very strong Hispanic population, and they're enriched with genetic predispositions to Nafld severity in the form of PNPLA3. But you know, the quote I use for Texas, it's where genetics meet tortillas, because this is really a perfect storm of dietary issues that predispose patients to fatty liver disease, and maybe this is where the crux of the matter lies. I mean, if you're in Europe and you're enriched for PNPLA3, but you eat a Mediterranean-based diet, then maybe you're not as predisposed to NASH and NASH severity as if you're in the southern part of the United States and you're eating, you know, again, a very enriched carbohydrate-based diet. So looking at this data, this is data that I've collected at Brooke Army Medical Center over the past almost 4.5 years now, and this is an interim analysis. We hope to be complete with the study in the next month and a half, and we'll have final data at EASL. We've enrolled over close to 800 patients prospectively. These are patients that present to the hospital for routine colon cancer screening. They otherwise are healthy individuals. And we ask them if they would like to learn their liver health at the time that they're getting their colonoscopy obtained. And we exclude patients that drink, you know, alcohol to the extent that puts them into the alcoholic liver disease category. We exclude patients that are on medicines that cause fatty liver, and we exclude patients that have a history of other liver disease. So then if they meet criteria, they then undergo extensive imaging, basically including FibroScan with CAP, MR elastography, MR PDFF, and multiparametric MRI. And so I think it gives us a very enriched, a very robust set of prospectively obtained data, albeit at a tertiary care center and albeit in a population of patients that are presenting for routine colon cancer screening, where the mean age was roughly 55 years of age. But having said that, we do see from this data set, using again the gold standard PDFF, 32% of patients have fatty liver, but more importantly, 17% of them have NASH. And again, this is much higher than what we would see in the published literature. But I think there's a couple caveats from this when we look at the data a little bit closer, Piat, that are worth mentioning to the group, and that is focusing on diabetics. If you're a diabetic, 71% have fatty liver. That's in keeping with the published literature. But more importantly, almost half of them have NASH. And so I think that's a number that people tend to overlook a bit. And getting back to the prior meta-analysis slide that Rich looked at in CGH, this data tends to agree with that. where you see Hispanic prevalence, being a little bit more common than Caucasians and Blacks. So when we shift back to my data slide, you see that illustrated again here, where 57% of Hispanics had fatty liver, 29% of Caucasians, and 20% of African Americans. But where we drill into the data a little bit more than what you saw in the meta-analysis is each of these patients had a liver biopsy. And so we see the prevalence of Nash in Hispanics being 34%, that of Caucasians being 15%, but African Americans, only 3% of those guys, three out of 3% actually had Nash. So again, I think that may provide some telling points when we look at the differences in epidemiology between Europe and the U.S. And then if you look at Asia, what we didn't mention and what was not highlighted in the other publications, and again, my data set here is not too robust. There's only 20 Asian patients, but the prevalence of fatty liver was 30% and the prevalence of Nash 20%. But I think, ultimately this paints an epidemiologic picture that might be slightly different, except for potentially countries like Spain and Portugal that have an enriched Hispanic heritage, and maybe their diet might be more in line with what you would see in the southern part of the U.S.
Thanks, Stephen. This is great insight into epidemiology. But what do you think, and what your experience tells you, what drives these Hispanic patients in the U.S. to actually get involved in clinical trials? Is there something there that we can leverage or change in Europe?
Well, I think the Hispanic community in the south of Texas is a very close-knit community. The families are very large, and they tend to share health information very readily between them. And so, it's interesting when we see patients that are screening for clinical trials, they'll often mention that they have a sister or a brother or a cousin or an aunt that has fatty liver, and can they bring them in and get them screened for a study as well? So I think we don't see that necessarily with Caucasians or certainly with African Americans. So I think it's something unique to the Hispanic heritage and the culture that they have a kind of a closer network of sharing health information and encouraging their relatives to seek health care. So I'm not sure if that's the way it is in Europe at all. It might be, but I'm not sure that's the case.
And switching gears a little bit from patient's perspective to physician's perspective, if we look at patient management paradigms in Europe and US, do they differ? Do they substantiate these recruitment rate discrepancies that we noted?
Yeah, I think a little bit. Probably, it's amazing how quickly these practice guidance documents go out of date. So this one is the European practice guidance published in 2016. And it talks about if you have metabolic risk factors, get an ultrasound. If there's fatty liver and there's abnormal liver enzymes, then refer those patients to specialty care for consideration and further workup. And then obviously, if there are normal liver enzymes, then obtain serum fibrosis markers. And if those are high, refer patients. I think where we differ a little bit now is a couple things. One, both in Europe and I think in the U.S., we've added in a level of complexity here where we go to FibroScan as a non-invasive assessment, maybe even at the point where you have metabolic risk factors present. And if it's normal, the FibroScan is normal, the negative predictive value of that test is 95%, which means there's really nothing else to do. And that's what I do in my clinical practice is basically I use it as a sixth vital sign. And I saw 14 patients yesterday. All of my new patients had a FibroScan before I saw them. those that were normal that was a completely different conversation about fatty liver disease and management, i.e. lifestyle modification, versus those that had an abnormal FibroScan, which basically told me that we needed to do more workup. And those were patients that we had a discussion about a liver biopsy. We had a discussion about clinical trials. And basically, where would you do the biopsy? Would you do it in clinic where you have a copay, or would you consider doing a liver biopsy under the auspices of research where you don't have a copay? And I think that differs in the US from Europe. In Europe, patients there's been a culture shift, a paradigm shift, if you will. I think this stems from the hepatitis C world, where people are a little bit more hesitant to jump to liver biopsy. They're a little bit more willing to accept what the FibroScan shows them, and basically put into place a treatment paradigm that doesn't involve liver biopsy. And as you know, with clinical trials, often we need a baseline liver biopsy, we need an end of treatment biopsy, and in our phase 3 trials, we even need a third biopsy often at the 5-year mark when we look for hard endpoints and progression to cirrhosis. So just the way I've thought about this is, in the US, I think there's still a sense that FibroScan is helpful, but it's not the definitive diagnosis. In other words, we use it as a screening tool, but not as a diagnostic, where I think in Europe, it's often used as a diagnostic and treatment paradigms are put into place based on the results of that scan. So we're basically having to ask the physicians and the patients to go backwards in time and actually do the liver biopsy to qualify for clinical trials. So at least for clinical trials in NASH that involve histology, I think there's been a little bit of a maybe a delay in European countries enrolling clinical trials as quickly because of that biopsy related issue.
Yeah, and we certainly see this when it comes to convincing patients to come to studies. The experience of our investigators in Europe do tell us this as well. So switching to gears a little bit now to the biomarkers in early NASH clinical trials. I just wanted to pull up this table, which is the listing the biomarkers which showed early, which I arbitrarily defined as less than 24 weeks, yet statistically significant response versus placebo in NASH trials. So this list is fairly long, robust. There are some missing biomarkers. We can talk about this in a minute. But I think at this time we can be fairly confident of responsiveness and overall validation, if you like, of a number of non-invasive biomarkers, both imaging and liquid, based on this review. But which ones to use though? Are there any selected biomarkers you believe are more important than others? You presented this data on the next slide recently.
Yeah, so this is fascinating to me. This is really where I think we took a quantum leap in our understanding of NASH as it relates to non-invasive biomarkers. And as a result of that quantum leap, I think if you are in the business of drug development for NASH and you even partially paid attention to the data, you're going to be excited to know that there is a strong correlation between improvement in biomarkers and improvement in histology, at least from what we were able to glean from a uncontrolled open label trial that NGM presented. And that's the data that we see in this slide. And I think that the take home point is, NGM 282 is a basically a recombinant FGF19 analog that has relative pleiotropic effects, but mainly focused on the metabolic aspects of fatty liver disease. So you see a couple things. Number one, what we learned from, and this is a little bit of historical context, what we learned from EASL was that bile acids are playing a central role in this disease. Bile acids are linked to NASH severity. They're also linked to fibrosis. And by inhibiting bile acid formation, which is what FGF19 does, or NGM282, you suppress bile acids significantly. And so what's not listed on this slide, and I think is important, is you need to have a measure of target engagement if you can. If there's a marker that you can measure that says your drug is having a robust effect on the target, then that's informative. And FGF19 had that in the form of C4, and it was almost completely reduced to zero. So having that, then looking at MRI PDFF, which is kind of the gold standard for the measurement of fat in the liver, along with inflammatory biomarkers, ALT, and liver inflammation fibrosis score, corrected T1, which is a number we get from multiparametric MRI, and then the non-invasive fibrosis biomarkers, PRO-C3 and ELF. What you see here is that all of those were moving in the right direction. And so the take-home point for me is the more of these parameters, fat, inflammation, and fibrosis biomarkers that are moving in the right direction concurrently, then I think what it shows us in the last two lines of this slide with the Nafld activity score and the fibrosis average baseline to week 12 fibrosis change is that you can begin to infer that you potentially would see a histopathologic benefit if you're seeing improvements. And I would like to highlight in addition that in this particular trial, 60% of patients completely normalized liver fat in 12 weeks. 60% of them completely normalized ALT in 12 weeks. And that ultimately led to the changes that you see in histopathology. So, there are some markers that we didn't show here that I think we'll comment on in the next couple of slides. But I think the take-home point is twofold. Number one, you can improve fibrosis without being directly anti-fibrotic, and you can gain a clue that you might be having an effect on histopathology if you see significant drops in PRO-C3 and ELF. And the same goes for NAS improvement if you see changes in PDFF and ALT.
Mm-hmm. And what about the other biomarkers that we have been seeing used in clinical research so often, such as CAP or FibroScan or NFS, FIB4? What is their utility in trials? Are they responsive enough, or would you see other roles for them?
Yeah, my own personal opinion with CAP, FibroScan, FIB4, and Nafld fibrosis score is that those are best used as a pre-screen strategy. And at least for for CAP, we have pretty good data, although it's still evolving, that CAP does correlate with PDFF and does correlate with the grade of steatosis. It varies a little bit more. It's not quite as precise as MRI PDFF, but let's say you're designing a clinical trial and you include PDFFs of greater than 10%. Well, MRI is expensive. You don't want to screen fail patients on MRI. So by the time you get through signing consent, you get the blood work, and you order the MRI, you want to pretty much screen fail all those patients that are going to screen fail before you get to MRI. So the strategy I employ with CAP is I use a CAP in a pre-screen just like the kilopascal score, to inform whether or not that patient is likely to have an MRI PDFF that qualifies. So essentially, for a MRI PDFF of 10 or more, you kind of want a CAP score of greater than or equal to 300. That gives you the best sensitivity and specificity, or what we call Uden's index, for predicting fat content greater than 10%. And the same thing with KPA. It depends on if I'm looking for an F1 or population, F2 population. I'll use different cut points on the KPA to help enrich the probability that that patient will qualify. Because again, the number one reason for screen fail on liver biopsy is there's Nash, but there's no fibrosis. So you want to try to eliminate the non-fibrotic Nash patients, and you don't do that with just the KPA. You also want to bring in what I like, either a Nafld fibrosis score, FIB4, or quite frankly, I use AST, which is a big component of Nafld fibrosis score and FIB4, but it's much simpler to use. And so I've tried to put together a cutoff of, a specific KPA number with an AST number when I start to look at fibrosis stages. So that's the concept, but between CAP and VCT, Nafld fibrosis score, and FIB4.
And what we often see in early NASH trials is the use, and we see this more and more, the use of rather sophisticated technologies as biomarkers. Are they limited by their availability? Is the feasibility of studies and how is study design affected by the availability of these technologies in your experience?
Yes, unfortunately they are. Here you show a graph of MR elastography, and while there, I think you had mentioned a couple of days ago, there may be more than, on the website, more than 1,000 MREs installed around the world. Is that a right number?
That's the number from the manufacturer, yes.
Yeah. The problem is that hasn't trickled down to the sites that are high enrolling, NASH clinical trial sites. We actually see that very plainly. I lead the work package 5 for Litmus Consortium, which is kind of this European consortium that's been put together by the IMI to basically develop biomarkers for NASH. It's not just based on imaging. There are a lot of wet biomarkers that are being assessed. But for me, leading the imaging work package, it's been an eye-opener for me to see the few sites that treat NASH patients, that enroll NASH trials, that have MR elastography. In fact, I think it's only 8 of our sites in Europe that have MR elastography. And the two main sites for the Litmus Consortium are Newcastle in the UK and Oxford University, and neither one of those have MR elastography. And working with my site network here in the US, that's one of the things we're trying to do, is to get all of our site networks up and running with a full complement of MR technologies, to include multiparametric MRI and MR elastography. And one of the limits that we're seeing with MR elastography is the significant cost it takes to upgrade. Whether you look at Siemens, GE, or Philips, not all MRI scanners are created equal. Some are very old, some are newer, and some require a significant hardware and software upgrade. But ultimately, the average cost to upgrade a unit, whether it's in Europe or whether it's in the US, is running about $100,000 to upgrade to MR. And quite frankly, without a reimbursable CPT code yet in the US, radiology centers are very hesitant to invest that sort of capital to upgrade unless the sites can somehow show them a return on investment through research. So it's been a bit of a challenge to bring people up to speed on that. Having said that, I do think we're a little limited on, if you have an antifibrotic drug, or you have a metabolic drug and you're wanting to assess whether non-invasively there's been improvement in fibrosis. You saw preliminary data from NGM on ELF and PRO-C3 as two wet biomarkers, but really MR elastography is the best at showing us what the liver looks like from a fibrosis perspective. And so this test, I think, has significant utility as a non-invasive assessment marker. But two things. Number one, what we've learned from the data that's been presented so far with MRE is that potentially, if you defat the liver quickly, you may actually, in the short term, worsen the MR elastography, just because one of the thoughts is you're condensing the collagen because there's no fat separating the collagen anymore. And so you're taking a very big liver, defatting it, shrinking the liver down, and so basically you're consolidating the collagen. And that's one theory as to why maybe the MR worsens in a short period of 12 weeks or so. If you don't defat the liver, maybe that's not an issue with MR, but most people believe that either you need a very robust antifibrotic or you need more time, like maybe 6 months, to show MR elastography changes. So more to come with MRE. I think it does have utility in the right setting, but we do need to do a better job of getting sites up and running with that capability.
Yeah, and certainly this is ours. This role is ours to help investigators setting up these technologies. Shifting gears a little bit now to study endpoints and their selection as we move with a successful drug development. How these have changed and evolved over the last years.
Yeah, and I think, this is a nice summary to kind of speak to what we've already addressed, and that is, what are some of these endpoints for outcome measures in NASH? And I think, it depends on the phase of development, and we'll get to that in the next slide. But if you focus on clinical parameters, and this is ultimately what the FDA wants, is, does your drug affect how a patient feels, functions, or survives? And whether you look at progression to cirrhosis, all-cause mortality, or liver-related mortality, there are measures that we can use to assess that, and I have those outlined there. When we look at metabolic, inflammatory, and fibrotic changes, again, you can use these non-invasive parameters to assess, mainly focused, first of all, on fat. Really, PDFF's the gold standard there. You can also do multiparametric MRI. When you do multiparametric MRI, at least through liver multiscan, you get PDFF. I think that's often overlooked, but you get the fat quantification at the same time that you're getting the liver inflammation fibrosis score or the corrected T1. You can also use CAP, and I've talked about how I would use that. The one thing that is a little challenging, we've talked about fibrosis endpoints as well, but the inflammatory change, we don't have very good non-invasive assessments of that in the form of wet biomarkers. All that we really have is ALT. I think people try to look at CK18, the fragments of CK18, the 30 and the 65. They look at TNF alpha. They look at IL-1, IL-6, fibrinogen, and ultimately, I think those are not very sensitive or specific for inflammation. So if I were designing a trial and I had an anti-inflammatory or I had a drug that was pleiotropic and in some ways I thought might have an impact on inflammation, my main thing I would look at would be ALT and AST, as well as corrected T1. So maybe we can go to the next slide and talk briefly about different phases of development. I published this data a couple months ago in Journal of Hepatology, and I tried to just lay it out, spell it out in crayon for those people in drug development to say, look, this has kind of been bounced off the regulatory agencies kind of trial and error type thing. And this is at least in February where we stood, and I think it's still for the most part true. At the top on the left is phase 2A, then phase 2B, phase 3, and adaptive design, and really looking at what the focus of each stage of development is, what the current timelines are, whether or not a liver biopsy is needed, and then taking those three different components, metabolic, inflammatory, and fibrosis, and beginning to see what you can measure. And I think some of the takeaways from the liver meeting, just to summarize, would be if you're designing an early phase trial, like a phase 2A, the goal is not liver biopsy. The goal is, quite frankly, is your drug safe? And number two, do you have evidence of target engagement? And do we begin to see non-invasive measures moving in the right direction? Again, 2 is more than 1. 2 is better than 1. You want to see more than one thing moving in the right direction. So if your drug is, let's say it's a predominant drug that affects fat, maybe it's an ACC inhibitor, maybe it's a thyroid hormone receptor beta agonist. We learned from Madrigal about that drug at the meeting. Then you're going to want to use PDFF because that's a very simple, well-validated measure. But you want to see something else move in the right direction with PDFF. And really, ALT is kind of the easy one to look at there. But you could also begin to look at some of the fibrosis biomarkers. Now, next would be a phase 2B. And in phase 2B, obviously, you want to see histology. The agencies are very cautious right now. They want to see histology prior to moving to phase 3. And they also want to see histology in a control population. You could do an adaptive design where you do a phase 2A/2B, like Madrigal has done, where you do a 12-week primary endpoint assessing non-invasives, but you have a tail where patients continue on drug for a predefined period of time, and then you repeat a liver biopsy. So now you have paired histology. It's a secondary endpoint, but it does give you data in a control population and in a treated population that potentially is far enough along that may inform the development of your phase 3 program. I think that was the main points I wanted to get out from this slide. Piat, if you had any other comments or questions that we could address, or open to questions to the audience.
So thank you, Dr. Harrison. That concludes our planned presentation for today. So this does open up our official Q&A session. Remember that if you do have a question to send through, to use the chat feature. We do have a couple of questions so far. And this one kind of piggybacks on what was just discussed about having the histology incorporated into the protocols. But with early NASH trial designs without a confirmatory liver biopsy at baseline, how would you approach patient selection in such trials?
Yeah, I would. That's certainly a hot topic right now, and I would really try to aim for patients that are likely to be the same types of patients that you would enroll in a phase 2B where you had histology. So if I were going to do a non-invasive assessment of patients to enroll in a phase 2A, I would focus on probably MR, multiparametric MRI with corrected T1 or lift score. I would take people, I mean, there's data presented at EASL showing lifts of 2.5 or greater represent a NASH population with at least F1 disease. Or you could take an MR elastography, and at that point I would use 2.5, 2.6 on my KPA as well. And obviously I would make sure that they had a decent amount of fat, particularly if you were aiming, if you're a metabolic drug, I'd make sure they had more than 10% PDFF, because when you do that, the mean PDFF turns out to be somewhere between 18 and 20%, 17 and 20%, something like that. And that gives you a nice cushion to show efficacy of your drug over time.
Okay, thank you. So with the patient enrichment strategy or the pre-screening strategy that you discussed a little earlier, what screen failure rate based on liver biopsies do you experience at Pinnacle?
Well, it's better. It's getting better. It's not where I'd like it to be yet. But roughly, we've been able to take screen fail rates from 60% on liver biopsy down to around 40%. I still think there's room for improvement there to the 30% range, with a little bit tightening up those numbers a bit. But this strategy works, and it works for a couple reasons. Number one, the coordinators don't get discouraged as often, right? If you have a lot of screen fails, the coordinators get discouraged, the patients get discouraged, the PI gets discouraged, and ultimately people, just from human nature, begin to look for other clinical trials for their patients. So everybody wants to be successful. The patients want to be successful. They don't want to be to get the phone call that says they screen failed. Nobody wants to be a failure. So ultimately, it's been tried. It's been true. Whether we've enrolled a very large phase 2b trial recently using this pre-screen strategy. I presented some of that data at Park City in January at Nash that showed a 20% reduction in screen fail rate from liver biopsies, and what that led to as far as cost savings and enrolling the trial quicker. So I think it's possible. I think we're still not finished product yet, but the tests that I use to do that are twofold. One, a FibroScan, and number two, an AST. I think what we tried to do early on was just a FibroScan, and if you look at that in isolation, we still get way too many false positives. But when you combine that with a serum AST, not ALT, but AST, and you bring the cutoff to 30 for an F1 and 40 for an F2, you dramatically drop your screen fail rate. Now, you do that at the expense of leaving behind some patients that might otherwise qualify for this study. So you have to be willing to accept that. But if you are working with a group of high enrolling sites, you're okay with that, because ultimately you're still going to get a ton of screened patients, but you're going to be highly selective in those screens. And so ultimately, for a pharma company or a biotech company, you're not paying for a lot of screen fails, and at the same time, you're enrolling your trial quicker. And just a comment on screen fails. I mean, it's become more and more common that companies do pay for those screen fails, not just a percentage of them. So, in designing your trials and your budgets, you want to make sure you take that into consideration. So it's, again, all the more reason to enhance your pool of patients to minimize the screen fail rate.
One other question to kind of piggyback on what you just said about the AST levels for an F1 or an F2 patient. What are the KPA cutoffs specifically for F1 or F2 that you're looking for?
Well, it's a little different in the U.S. than it is outside the U.S. For us, it's interesting. There's not a lot of published data on CAP and FibroScan and Nafld patients in the U.S. There's a nice paper by Elliot Tapper published in the American Journal of Gastroenterology using the M probe only in a small number of patients, I think less than 100, and that cutoff was 7.9. But I think it's actually higher than that. In my own experience from the data we have at Brooke Army that I presented to you earlier on the prevalence numbers, again, we obtained all that imaging data and correlated it to biopsy. Plus, we've enrolled several hundred patients in clinical trials that have had FibroScan and liver biopsy. And at least for me in South Texas, what works best is a KPA of 8.5 and an AST of 30 for an F1 or greater population, and a KPA of 10 and an AST of 40 for an F2 or greater population. And again, I can't emphasize enough, you can't look at those in sequence. They need to be looked at in parallel and together.
Okay, thank you. Moving back just to some questions on trial design, would you support an early proof of concept trial in an anticipated NASH indication run in patients with Nafld? In other words, only steatosis endpoints?
You can do that. I think my bias is it's shaped by having been at the FDA for several meetings, and they look at Nafld as a different patient, as a different group than Nash. And ideally, I think they would like to see in early phase that you have a signal in Nash patients, the same ones that you would consider enrolling in a histology-based study. Having said that, they have never said you can't look at just Naflds, but you're going at risk, I think, a bit to do that.
Okay. So I think this is the last call for any questions. We don't see any more that have come through. Any final closing remarks, Dr. Harrison?
No, I think we've covered quite a bit. I want to say thank you for your attention today, and I think, again, stay tuned, because in the next 6 months leading up to AASLD, there will be significantly more data coming out that helps inform us about non-invasive strategies and how we can design clinical trials to optimize those strategies to get the answers we need to move drugs through safely and quickly into the Nash field.
Yes, thank you very much, Stephen. Thank you, everyone.
Yes, thank you for your time today.
All right. Well,
thank you.
Thank you, Ms. Harness. Thank you, Dr. Shievsky and Dr. Harrison, for that presentation and the Q&A session. That was fantastic. If you do have any further questions, you can contact Medpace via this email showing on your screen right now. That's info@medpace.com. And thanks everyone for participating in this webinar. You'll be receiving a follow-up email from X-Talks with access to the recorded archive for this event. And a survey window will be popping up on your screen shortly. Your participation is appreciated, as it'll help us improve our further webinars. Now, I'm just about to send you a link in your chat box. You can tweet about the webinar using that link, so I encourage you to do so. Big thank you to Dr. Stephen Harrison, Ms. Jennifer Harness, and Dr. Piatrashievsky. We hope that you found this webinar informative today. Have a great day, everyone.